Hello and welcome to this webinar titled Subvisible Particles Matter, Developments in Regulations and Low-Volume Methods, hosted by Biopharma Asia magazine and presented by Dr. Satish Singh, Head of Drug Product Process Development in the Drug Product Services Group at Lonza Pharma and Biotech. My name is Orsoya Balog and I will be the moderator. Now, please allow me to introduce our presenter, Dr. Satish Singh, Head of Drug Product Process Development at Lonza Pharma and Biotech. As a leader for contract development and manufacturing, Lonza Pharma and Biotech is recognized for reliable, high-quality services, global capacity, innovative technology platforms, and extensive experience. Lonza's broad capabilities span across biologics, small molecules, bioconjugates, and cell and gene therapies. Lonza Pharma and Biotech manages projects from preclinical stage through to commercialization, and its expertise covers both drug substance and drug product. Lonza Pharma and Biotech believe that the best outcome for its customers and their patients can only come as a successful collaboration. Dr. Satish Singh is the Head of Drug Product Process Development in the Drug Product Services Group at Lonza Pharma and Biotech, where he is leading the drug process development and characterization capabilities. Prior to joining Lonza in September 2016, Satish was Research Fellow and Group Leader at Pfizer's Biotherapeutics Pharmaceutical R&D Unit. Satish has more than 25 years' experience in the industry in product development activities and is an internationally recognized expert in all aspects of biopharmaceutical drug product, biologics, vaccines development. His experience includes ophthalmics and parenterals encompassing primarily biologics but also small molecules, and he has been involved in the submission of several BLAs and INDs. Satish has a strong technical background complemented by a deep understanding of regulatory challenges and the evolving regulatory landscape, and has more than 70 scientific publications in the field. He is involved in various professional groups through the AAPS as well as the USP, and was a member of the expert panel that wrote USP 787 and 1787. His research interests also include the investigation of impact of freezing processes on proteins, use of computational approaches to mitigate developability issues, and aggregation and immunogenicity. He obtained his BTech from the Indian Institute of Technology, New Delhi, and a Master of Science and PhD in Chemical Engineering from Kansas State University. He holds the position of adjunct professor at the Department of Physical Pharmaceutical Chemistry at Uppsala University, Sweden. Welcome, Dr. Singh.
Okay. Thank you, Celia. And thank you to the audience for joining me in this webcast. I'm going to talk about as indicated on the title, I'm going to talk about subvisible particulate matter and developments in the regulations, and particularly in the low volume methods, with my focus being on biotherapeutics, which is what has been actually driving a lot of the developments in the subvisible particulates area. There's a disclaimer from Lonza, and my the talk outline, I will briefly introduce the particulate matter topic, then talk about some of the regulatory trends in this area as indicated in the title, then cover USP 787 and small volume methods. I will obviously not go through the full chapter, but just cover some salient points of that chapter. A little bit of discussion on particulate strategy and analytical methodology. I have a set of slides at the end which covers particulates in biosimilars, is of a particular interest of mine, but I probably, in the interest of time, will not go through it, but the slides will be available for anybody to reference. So this is my abstract, and I again will cover the topics that I listed before, and now I can jump into the into the main part of the talk here. You know, I've been working on this question of subvisible particulate matter for almost 10 years now, and although a lot of things have been settled, there is still a lot of technical developments and still a lot of unknown questions, you know, and new questions that arise in this area. So it's a topic of evergreen interest. But let's look at what is subvisible particles and what are the pharmacopoeial requirements related to subvisible particles in the three major pharmacopoeia. And you will see that per the definitions in each of them, they are aligned, they are harmonized, there are minor differences in the wording, but they all refer to extraneous mobile undissolved particles. So this is particles that we do not want, so they're unintentionally present in the solution. So this relates, the chapters as they have been written originally relate to extraneous, undissolved, unwanted particles in our solution. So the intention is that parenteral solutions, parenteral products should be clean, as we would certainly like them to be if we inject them into ourselves or into people we care for. So the intention is that this is an important quality parameter of every parenteral product, that it should be free of these undesirable particles in solution. A related requirement, although we will not cover this in detail today, has to do with visible particles. Obviously, particles can be subvisible or barely visible to the eye, and then visible. And here, there are, again, the definitions are more or less aligned between the three major pharmacopoeia. We all know the term about the parenteral solution should be practically free from particles, or essentially free from particles, or in the Japanese pharmacopoeia, free from readily detectable foreign insoluble matters. So this all relates to having parenteral products free of particles. In the USP 790 tries to define this concept of essentially free based on AQL characteristics, based on AQL requirements. And then just for reference, the monoclonal antibodies chapter in the European Pharmacopoeia says liquid or reconstituted freeze-dried preparations are without visible particles unless otherwise justified and authorized. So in the original version of this chapter, actually it said are without visible particles. That means no particles at all. And so this was sort of not in alignment with the general chapter about injectables. And therefore this caveat was added, unless otherwise justified and authorized. But the principle is that these products should be free of particles as far as possible. So going back to the basics here, what are our particles like? And this chart sort of gives you a little bit about how we categorize particles. Here we have, 1 micron to 100 micron, and obviously this is a fuzzy boundary. We generally tend to call them subvisible particles, and then particles above 100 micron tend to be visible particles. Obviously this is not a strict boundary, a little bit dependent upon the type of particle, the probability of detection, etc. But for the purpose of discussions, this is a fairly reasonable cutoff. And then particles below 1 micron are so-called submicron particles. Then if we look at these, particles above 10 micron have been historically controlled, and these were in days prior to biologics. These were parenteral products where the risk was based upon foreign particles and their risk for causing capillary occlusion after being injected into the body. So the same risk still obviously exists for foreign particles, but then there are the protein particles, which is the subject of today's discussion. Are protein particles a capillary occlusion risk? We don't know, but protein particles tend to be flexible and gel-like and potentially have a smaller capillary occlusion risk, but that exists. But the bigger risk with protein particles has been outline as immunogenicity, the risk for causing an immune reaction against the primary product, against the active antibody. And that is the primary reason for control of protein particles or proteinaceous particles in our product. And then there is another imperative for us to look and control particles, is that particles are a good measure of process and product characteristics and process and product control. So whether there is an immunogenicity risk or a safety risk or not, that depends upon the product in question. But in general, a control of particles is a very strong measure of control of process and control of product characteristics. So from that perspective, it is important to understand the particles in our product. So if we go back and look at how we control the particles, if you look at protein aggregation, which is where the particles arise from, we often use size exclusion chromatography, and we have specifications on that. But size exclusion chromatography covers particles that are small in size, potentially down to 0.1 micron or below, because larger particles will get filtered out of the product, out of the column, and so will not be detected. And then we have 787 or 788 that covered particles above 10 micron and put specifications or limits on those. So then we had this, so there was this control gap. There was this gap between which proteinaceous particles were not being monitored or being controlled. And that was sort of the reason for the ongoing discussion, for the start of the discussion about protein particles. If you look at all the methods that were present, again, there was this measurement gap. There was, of course, size exclusion chromatography and other methods on the low side, and then our light obscuration, microflow imaging, and so on above the 2 micron sizes. And so there was this measurement gap, which has now been, with further developments in this area, with resonant mass measurements and other new, improved Coulter counter and so on, have tried to push the measurement gap towards smaller and smaller particles. But there is still a gap in which the physics prevents accurate detection and or measurement of particles. But that's the current status. So in summary, we have subvisible particles. The original concern was capillary occlusion. A new risk with therapeutic proteins is immunogenicity. And then we understand that with subvisible particles, there is a good way, it's a proper way to control, it's an important way to control our product, to control and characterize our product. One thing I would like to cover here is also the particles in parenterals, especially in the USP. There are 3 categories of particles that the USP defines, so-called extrinsic, intrinsic, and inherent. Extrinsic particles are particles that are foreign. They're not part of the product. Their source can be foreign or unexpected, such as fiber, rust, hair, plastic, insect parts, and so on. These would be sporadic. Hopefully the facility is in such that these do not become a common occurrence. They would unlikely to be present in all containers, but once they are present, they will be constant, and they will be, they will be additive in the sense that they add to the product, to the particle count in the product. But these could be a sterility risk. I mean, if you have insect parts and fibers coming in, this could potentially pose a sterility risk for the product and could indicate a lack of proper GMP controls. So these are truly particles that are foreign to the product. And then there is a set of particles called intrinsic particles that may not be foreign, but they may arise from the product or the package. The source could be the product, the package, and the processing conditions, and this could be identified. For example, glass, metal, rubber, silicone oil, etc. They again could be present in all containers, but they may not be present in all containers. Some of them could be, consider whether they are extrinsic or intrinsic. If you get a metal metal shaving from the filling line, is that an extrinsic particle or an intrinsic particle? So some of these definitions are difficult, but they could be additive or changing. If it's, for example, silicon oil, the levels or the type of particle could change over time. And then there are, with the protein particles, there is something we define as inherent, that is of the product or product related. Again, examples could be proteinaceous particles or silicon oil, or if excipients degrade in the product. For example, lately with polysorbate degradation, you could have particles arising because of that. So these could likely be present in all containers. We would expect this to be constant in all containers, but that's not necessarily the case always. These could be constant or changing, and in my mind, this indicates a need for improved control or better product development of the product. And in all these three cases, patient safety impact has to be considered. I must note that these are USP definitions only, and they're not defined in Pharmacopoeia, in the European or the Japanese Pharmacopoeia. And the consideration about risk from these particles, whether inherent, intrinsic, or extrinsic, is not exactly clear. It is not always clear that extrinsic particles, let's say if there is one extrinsic particle in the product, is that more of a risk than 100 proteinaceous particles, or the other way around? This is not always clear which way does the risk go. Furthermore, the definitions are challenging and often difficult to assign, especially the smaller the particle. It's not always easy to know unless you do a fairly extensive exercise of identification of particles as to what type of particles do we have. Particles can be, obviously, in a product could be a mixture of these, and as I mentioned before, inherent particles, that is proteinaceous particles, could in some instance pose an even higher patient safety risk than the other two. For example, if it's a proteinaceous particle that can cause an immunogenicity risk, you may have a greater risk with inherent than with extrinsic particles. So while the definition is, while this categorization is good and useful to have, it is difficult to use in practical circumstances. And here is some just typical images for a high concentration product from a syringe. The type of particles one could see, you have this typical silicon oil droplet type of particles with this, spherical round shape, and then proteinaceous particles that are, gel-like and amorphous in structure. And in this particular image set, you can see that there are some particles which actually comprise both. There are silicon oil particles that are trapped in these proteinaceous gels. So you can have this combination of particles also. And you can see that the proteinaceous particles present a wide and diverse type of morphologies. So let's look at some of the regulatory expectations or regulatory trends. And I'm not going to go cover all the text in here in these slides that were taken from Susan Krishna's presentation at Breckenridge workshop a few years back. Just cover some highlights. So here's the thing about subvisible particles above 10 and 25 micron. She writes that depending upon the clinical experience and manufacturing history, it may be necessary to establish acceptance criteria for subvisible particles that are lower than the standard USP recommendation. And this is probably not an uncommon occurrence, that at the beginning, at the approval stage, you may have normal, 6,000 and 600 limits, but as with experience, these limits may be required to be tightened. Then for particles between 2 and 10 microns, FDA is requesting that subvisible particles between 2 and 10 microns be studied using quantitative methods. They do not recommend the use of a specific method. It's up to the sponsor or the company to decide which method works best for their product. They do recommend that orthogonal methods or additional methods be used to validate the. I shouldn't use validation in the GMP sense, but anyway, to validate the primary method to make sure that you are seeing what you expect to see, and that your numbers are in the range, are true numbers. And then also within the particle size range between 2 and 10 micron, they expect the particles should be characterized to inform method development and selection, risk assessment, and specification setting. So maybe at licensure, you may not have enough data to set specifications, but potentially, post-licensure, as opposed to post-marketing commitment, there may be requirement to put specifications in the 2 to 10 micron range also. And these would be product-specific specifications. Again, for the 2 to 10 micron, they expect force degradation, stressed and accelerated temperature studies to understand the causes, to understand the risk for when these kind of particles could form. And based upon that, they expect the sponsors to propose an appropriate control strategy to minimize the formation of these particles. For example, if shipping causes a lot of particle generation, then figure out a way to improve the shipping conditions so that you don't form particles. So that is the basic premise behind this risk assessment and understanding of what causes what are the risk factors for your product in terms of subvisible particles. And setting specifications, especially in this range, they do not have pre-established limits, and so the limits will have to be based upon your specific product, but obviously as low as possible. For the sub-micron range between 0.2 micron and 2 micron, they expect to have the products characterized, to understand, and the qualitative methods to characterize subvisible particles in that size range. It is understood that quantification is not as robust in this particular size range, so they want qualitative measures of what is the status of your particle count in this sub-micron particle size range. And finally, as Rukman put it, you really have to know your product. I mean, the bottom line is you have to characterize your product over the product life cycle, from early stage to late stage, and as the product reaches approval or licensure, you should have an extensive understanding of the particle history, the particle risks about your product. And this obviously holds true not just for particles, but for other characteristics of the product. And then in this slide, again taken from Rukman's slides, they expect a staged approach towards particle characterization. In early stages, you want to understand particles that are present at release and stability. The pharmacopoeia standards are acceptable. In late stages, this becomes part of product characterization. You need to do a risk assessment, understand which conditions cause particle formation and what is their impact on the safety and efficacy of the product. So that determines the control strategy that you would put into place for the subvisible particles. And during licensure, they expect this information to be provided. Analytical methods should be qualified or validated, and particle types should be described. Okay, so now let's cover USP 787 a little bit. So this USP 787 has been in the US Pharmacopoeia since August 2014. It was put out for drafts in 2012, 2013, and the rationale for this chapter was to address the gaps in 788, the main subvisible particle chapter, with regards to testing of therapeutic protein injections for subvisible particulate matter. Some discussions at the USP in a conference in 2010 actually led to the creation of the team that wrote this chapter. This chapter is obviously not harmonized, but it was enacted to enable quick action between 2010 and 2014, which is actually pretty fast for Pharmacopoeia, by Pharmacopoeia standards for creating a chapter. Applicability was biotechnology-derived articles defined in 1045 that infusion preparations as well as natural source therapeutic protein injections. So some salient aspects of 787, and I will not go through every point here, but the chapter allowed sponsors to use either 788 or 787. It allowed alternative analytical methods with adequately developed particulate limits. So that means if you use something other than light obscuration, you would have to develop your own limits. But the alternative methods were not specified, obviously. But if you used alternative methods, for example, flow imaging microscopy, you could not simply use the same limits as light obscuration. The limits would have to be developed for that particular method. And it also allowed quantitative dilution if necessary, considering that some of the protein products were high concentration products. You could use dilution if required. But appropriate measures of verification were required in this case. It suggested that product samples should be tested in a manner that most represents delivery or use. For example, if you were a syringe product, it should be expelled and not poured out from the back to measure particulate count. The most important part was allowing smaller test volumes down to 0.2 milliliters and allowing individual units to be tested, obviously with the statistical sampling criteria in place. It clarified that for lyophilized products, reconstitution with the specified diluent must be done. The diluent must be tested. However, diluent counts could not be subtracted, so you could not discount them. And then it informed about, eliminating gas bubbles and avoiding sonication of protein solutions in that case. And then about the limits, the historical limits were maintained, although it acknowledged that setting up limits is most appropriately done on a case-by-case basis during development and licensure. But the limits derived from 788 were maintained, but should be considered provisional. That means post-licensure you may have to put new limits. An additional requirement was put into place for large volume injectables so that the total particulate load would not exceed 6,600. And this limit is now under discussion in the Pharmacopoeia discussion group. And there was a specific statement that limits apply to the filtrates for products that require filtration just prior to administration. With that, I'd like to sort of just my own personal opinion about aspects of 787. Dilution to analyze can be a tricky aspect. If you have high concentration product, it is convenient to dilute and measure, but you have to demonstrate that there is no or minimal change in count. So this should be used with care, and the diluent must be identified properly. If you use WFI, which is convenient, you may have a situation where you form particles because the appropriate surfactant and stabilizers are lost in the solution. So we have to be careful when you're doing dilution. Another thing, although the chapter allows small volumes to be used, you should use an adequate volume where it is available. So use a reasonable test aliquot volume, because accuracy of counting accuracy of counting increases when you have more counts. So the larger the volume you're counting, the more counts you will have, and therefore more statistically significant numbers you will obtain. You will have less variability. And another thing, although both light obscuration and membrane microscopy are mentioned in the chapter does say that membrane microscopy is not suitable to count proteinaceous particles, partly because the particles are gel-like and often not readily visible on the membrane. A frequently asked question is, can I use membrane microscopy if I fail light obscuration? And this is, it's possible, but this should be used more as a troubleshooting tool than a compliance test. You cannot say I failed LO light obscuration, so I'll use membrane microscopy and voila, I'm in compliance and I can release the product. Preferably you must define the method that you're going to use a priori, and For example, if you have a lot of silicon oil particles that get counted in light obscuration and not in membrane microscopy, it's possible that you can use membrane microscopy, but you cannot start with light obscuration and go to membrane microscopy in a routine manner. You cannot jump between methods for compliance purposes. Okay, some other thoughts. Obviously, 787 is not a harmonized chapter, so it sometimes creates confusion as to which method to use, since it's not valid in all jurisdictions. Obviously, if you're developing an intravitreal product with a fill volume of 0.1 milliliters, trying to do a large volume method such as it with 788 requiring 25 milliliters of solution can be a nightmare, especially if you have to pass 789. But in principle, all of the pharmacopoeia allow different or smaller volumes to be used, but the method has to be qualified and validated as with any other QC method. So even if you were going to the Japan or to Europe, you could use small volumes, you just would have to validate and qualify the method. However, the Japanese Pharmacopoeia has released a draft chapter 6.17 specifically directed towards particulate matter for therapeutic protein injections. And there are efforts for harmonization underway in the pharmacopoeia discussion group. So just a couple of points around the Japanese Pharmacopoeia and the draft chapter 6.17, which probably will be finalized next year. It has quite a few similarities to 787. It allows test volumes between 1 to 5 ml to be accepted. You can reduce the test volume down to 0.2 ml, but you have to show its validity and proper sampling volume accuracy. It allows individual containers to be tested, and otherwise you would have to pool as would be expected. Just the same as in 787, if dilution is performed to do measurements, you have to demonstrate the validity of the dilution by demonstrating consistent results regardless of the dilution level. As is usual, the test specimen should be statistically sampled and representative, and the results should be correctly representative of the particles in the population. The limits are unchanged, the same limits as in 6.07 or in the 6,600 for the 10 and the 20 micron. Actually, this chapter completely eliminates membrane microscopy. It's not even referenced. And as with 787 and 788, sponsors can choose to use 6.07 or 6.17. So there is a fair degree of similarity with chapter 787 in this case. So along with 787, there was a 1787 informational chapter that was written to create a bit more awareness about subvisible particles in therapeutic proteins. It gives strategies for measuring and characterizing proteinaceous particles in the 2 to 100 micron range, and the objective was elimination, characterization, root cause analysis, and non-conformance investigations. So by characterizing the particles, a number of objectives can be achieved. 787 does not provide a recipe about which methods to use when, but it proposes characterization of aggregates and proteinaceous particles in the aid of objectives based upon size, count, some studies on reversibility of particles, structure and conformation, chemical modification, morphology, and so on. So as you go, and you can, seem to have, the more information that you can get from about your particles, the more control you can have on the particles. And I'll come back to that slide. Seems like I have one slide in wrong order. But and as was mentioned in Rukman's slide earlier from the FDA, you have to have a comprehensive strategy for monitoring particles. In early stages, you will get your typical particle profile. You will characterize the aggregates and particles in clinical material. In later stages, you may use this in comparability. You may understand the relationship with the formulation. It can be used in enhanced characterization. And for post-marketing, it may be used as part of life cycle management. So what would you actually do? This the words in the previous slide are fine. In my mind, when in early stages, your monoparticles and potentially inherent particles. You would apply multiple techniques to the particles. You would do, obviously, you would select a lot release method. You would develop a lot release method. You would choose between, say, light microscopy. But the principle in my mind is that you want to test early and test often. You want to test the particles you're putting into the preclinical studies. You want to test particles you're putting into clinical studies, so that you have a picture of what you're putting into, what is the safety database that you're generating. Then in later stages, you want to study all types of particles that may end up in your product. You have your fill finish facility defined, so you want to study what's happening to your product in that fill finish facility. Again, multiple techniques would be applied. Understand the variability you're getting between vials, unit to unit variability. Examine the variability between units as well as pooled samples to decide, what volume of product you will use in your sample measurement. Establish your proper sampling plans, and if you have silicone oil particles, establish a silicone oil particle accommodation strategy. So these are some of the practical things that you may end up doing as part of your particulate strategy. And obviously, an important aspect of this is forensics, is to understand the source of the particles, and if you have new sources of particles arising, get to the bottom of them. And again, I'm thinking of this polysorbate degradation and impurities. If these start to show up late in your program, you really have some scrambling to do to figure out what's happening at that point in time. But it's important to understand what particles you have. So forensics and characterization of particles is important prior to licensure. And this is the same as it was in the FDA slides. You want to do stress testing and control strategies. Understand which kinds of stresses cause particle formation. Use these for method development and qualification, and then also use this for risk assessment and control strategy. Putting specifications, defining process parameters, hold times, handling and storage restrictions, in-process controls, process limits. Depending upon what causes particle formation or what causes degradation, you want to put the appropriate controls in place. And the controls can be in as specs or as process controls, and so on and so forth. So here's just a simple example of how this kind of information can be used. This is an example of a product format switch. So the first product was a lyophilized drug product in a vial. A new formulation was developed to go from the lyophilized drug product to a liquid drug product in a prefilled syringe. In the prefilled syringe, they found that there was much higher counts in this liquid based upon light obscuration method. There were much fewer particles in the lyo, and when it was measured with membrane microscopy, with flow microscopy, it showed a large number of spherical particles which were characteristics of silicone oil. So these could be discounted from the total counts, and the conclusion was made that the product change did not worsen the product quality characteristics. The higher counts in the liquid were due to silicone oil, and therefore by characterizing the particle you could accept this format change and the particle count change. Here's an example of a simple example of risk assessment. So in this case, impact of stress was studied after storage, freeze-thaw excursions, thermal excursions, transport, in-use infusion studies. and it was found that freeze-thaw excursions led to a significant increase in particle count. So in this case, additional instructions and precautions and monitoring was put into place as control strategy. For example, during shipping of the product, it was important to put in temperature monitors, to make sure that the product did not freeze. Instructions and studies were done to understand what would be done in case the product did freeze. What would be the action plan, and so on, so that that was the kind of control strategy that was put into place based upon the fact that freeze-thaw excursions led to a particle count increase. So a few slides on analytical methodology now. Remember we mentioned need for orthogonal methods. Why do we need orthogonal methods? Particles come in, various shapes and sizes and types. This slide from Dean Ripple and Mariana illustrates this. You could have particles that are debris. This debris could be coated with protein. You could have silicon oil particles coated with protein, and you could just have protein particles. So you can have a variety of populations. And if you measured simply particle size, and count, you would, they would have overlapping. All of these different types of particles could come in would fall on the same distribution. However, let's say you measured particle density as a characteristic, you'll find that these different particles separate themselves into different categories. And this is, the value of orthogonal methods that have different measurement principles, because you can do a better characterization of particles, a better understanding of the particle types, and any other characteristic based upon measurement by other methods other than what we are generally used to, which is light obscuration and light-based methods. So, are these, obviously the question is, are these truly orthogonal methods? But these are some of the methods other than light obscuration that are commonly, well, I shouldn't say commonly, but that could be used. Flow imaging microscopy is fairly common now. Laser diffraction, light scattering are methods that are ensemble methods that are not as informative, but do enable measurements to be done in small size ranges. Coulter counter or electrozone sensing, again, measures using conductivity of the sample, and that gives you a slightly different measurement. AFF is not that commonly used, but it's available. And then more from forensics purposes, electron microscopy based methods and spectroscopy methods. And more recently, nanoparticle tracking analysis and resonant mass measurements have come into play for measuring particles in the size ranges just below 1 micron. And this slide sort of, slightly older slide, but depicts some size ranges that are measured by some of these methods. The methods on the lower side, such as size exclusion and AFF and even AUC, measure particles or measure aggregates, but they quantify by mass. So when you do size exclusion chromatography, you get a percentage of protein which is aggregated, for example. Then when you look at the other side, in the 1 micron and the count, and these methods such as Coulter principle, the flow imaging, and so on, they quantify particles by count. So there is there's a disconnect between the two. There's one side there's measurement by mass, the other side there's measurement by counts, and this, that's just a fact that we have to deal with. So coming back to this thing about the characterization, there's a whole range of characterization that one can do in this, and the information content about the particles is obviously increases as you do more and more detailed characterization. However, obviously, the more detailed characterization you want to do, the fewer particles you can analyze, right? The sample size decreases, and obviously, the more resources you would have to spend to do this characterization. So it's a balance, but certainly as part of licensure as part of the BLA filing, some effort needs to be expended to understand your particles. So, again, going back to 2 to 10 micron range, you can size and count it. We can do a fair amount of characterization in the subvisible, in the submicron range. Sizing and counting is becoming possible. There's some ability to do classification. Maybe we can do reversibility studies. But there's another aspect of the particle sizes in the 0.2 to 2 micron range. There is limited experience. There is not so many publications as to what is the number of counts that we see in that range, size range. So often people will make measurements, and we can get numbers in the high 10,000s and maybe even higher, and we don't know what is normal. So in this range, we are still sort of working through. We don't know what is normal, what is present in our products at this point in time. So experience will as more and more experience is gained and more publications are made in this area, we will gather more information about this. There are some new developments in old techniques. I will not busy go through it, but these old techniques, there are advances being made in this particular area, and the slides are available for reference. So here are some of the main. This slide is too detailed and too hard to read, but these are some of the different tools that we have currently, most of us have in our toolbox: nanoparticle tracking analysis, resonant mass measurement, the Coulter counter, flow imaging, microscopy, and the light obscuration. And these have different size ranges over which they work. They have slightly different measurement principles, and the size ranges that and the type of data that they report is counts, although counts by, for example, converted from surface area versus converted from volume and so on. But one thing we do know is that with these different measurement methods, they don't allow, if let's say you measure by by light obscuration and then by resonant mass measurement or by NTA, they do not give you continuous size distributions. You cannot overlay the counts, the particle sizes from one method to the next method and so on, because of the discrete nature of their measurement. That's just I shouldn't say a limitation, but that's just the way these instruments work. So the refractive index of is an important parameter, although it's not something that we measure usually. Refractive index of a protein particle is important because of some of our measurements are made on the basis of light and refraction or contrast against the medium. Most protein monomers have refractive index in the range of 1.4 to 1.5, based upon matching index, matching density measurements. Matching refractive index measurements. In general, condensed or amorphous protein particles have refractive index indices around the 1.4 range, and spatially averaged, a proteinaceous particle will have thin and more dense areas. A spatially averaged range of 1.34 to 1.4 from quantitative phase imaging. This slide is from Dean Ripple from NIST. So particles, proteinaceous particles are with the indices in the 1.4, approximately 1.4 range. And this has important implications for when we make measurements, because obviously subvisible particles have different refractive indices, and in this slide, this is 5 micron silica particles in sucrose solutions. Depending upon the background, depending upon the background, the same particle could appear differently. So in this case, by changing the sucrose background from 0%, 20%, 40% sucrose solution, the same particle, in this case by flow imaging microscopy, look very different. And the same thing here in this case, measurements, unfortunately the MFI did not show up here, but by changing the refractive index of the medium, the particles, the detection ability of the particles changes between light obscuration and flow imaging microscopy, because these two methods, both of the methods work by contrast. So if the refractive index of the medium changes, the contrast reduces, and therefore the particles become so-called invisible. Here's a nice piece of work from by Zolz et al., Andre Have and company, who looked at various particles such as silica, polystyrene beads, proteinaceous particles generated by heating human serum albumin or by stirring a monoclonal antibody, and using sucrose concentration as a way to change the refractive index, which is given up here on the upper side of and by measurement by flow imaging and by light obscuration. And you can see that, for example, silica particles which have a refractive index of 1.4 are less impacted until the refractive index reaches that of the silica particle, and suddenly they become sort of invisible to the methods. Polystyrene beads, which have a fairly high refractive index of 1.59, are therefore not impacted by the change in the medium or the change in the background surface refractive index. Similarly, proteinaceous particles based on HSA and immunoglobulin are again impacted by the background refractive index, and the two methods, light obscuration and flow imaging, are impacted similarly, but obviously they have different counts because of the different sensitivities. And the same thing here is seen by protein concentration itself. In the previous slide it was sucrose concentration, here it is protein concentration, human serum albumin particles in human serum albumin solution, and again the effect due to refractive index is pretty apparent, that as the refractive index of the background increases, particles start to, if I want to say, disappear to the instrument. And this is shown also for a solution of NREL, which was diluted with its formulation buffer from its original concentration of 50 mg per ml down to 1 mg per ml, 2 mg per ml, and 50 mg per ml. And you can see in the 50 mg per ml, much less counts are detected compared to 1 mg per ml. So this is what I meant by when you do dilution, it has to be properly qualified. And we know that proteinaceous particles don't look like silicon oil particles. So that's just this picture here to emphasize. Another aspect that is important is the impact of sampling volume. When you're using light obscuration or flow imaging or Coulter counter, the volume tends to be these methods samples, they say up to 1 ml or larger volumes. But when we go down to resonant mass measurements or to nanoparticle tracking, these methods sample a tiny fraction. They sample in the microliters. So when you convert the numbers back to per milliliter or per container, you will have huge extrapolation factors that lead to large variabilities in the counts, partly because you're just extrapolating by hundreds or by millions of factor, and small variabilities in the measurement get expanded. So this is something to be aware of, and this is the reason why I mentioned that if you have an adequate amount of sample, use the largest sample that you can spare. Okay. And the same thing holds for number of test articles. If you measure individual test articles, in this case they measured 1 syringe, pooled 3 syringes, pooled 6 syringes, and pooled 10 syringes, and this is looking at the counts and the variability of the counts by flow imaging, I believe, in this case. Yeah, and if you measure individual containers and you measure a number of containers, you would tend to end up having larger variability in the counts as opposed to pooling. And this is obviously because even though we say we have the same uniform product, we know that from our experience that particle counts vary between containers, and in this case it was syringes. So with the silicone oil particles in the mix, you end up having larger variability when you measure individual containers as opposed to pooled containers. So this is just something to be aware of when we start measuring individual containers, that you have an appropriate statistical sampling plan so that you get a true measure, and you don't just measure one container and say that that is representative of the batch. Okay, so coming to some of my last slides, which decisions are being made with subvisible particles? In development, we are using them for formulation and process development. We also often use them as surrogate for visible particles. As we use them for setting controls and specifications. We use them for characterization and even diagnostic. And actually, if you look at some of the biosimilar filings, these are being used also as similarity establishment criteria. I will, just in the interest of time, skip some of my slides that relate to biosimilars. These are available in the slide deck that will be available on the website. And I'll just go to my last conclusion slide here. So in summary, control of particulate matter in parenterals is a quality imperative. This is an important criteria. Regulatory expectations have evolved from the original concern of capillary occlusion to immunogenicity risk. It requires characterization, monitoring, and control. Size ranges below 10 micron need to be controlled. Size ranges below 2 micron need to be characterized, and we need to understand the risk for particulate formation in our products and put in a control strategy. USP 787 enables small volume measurements. This chapter will be harmonized and potentially superseded, but elements will probably be incorporated into harmonized chapters. Multiple methods are required to fully characterize subparticulate population. Use the methods in a quantitative manner requires good understanding of the strengths and limitations of the methods and the technical ability. And extensive characterization can be resource and also skills intensive, and a phase appropriate strategy should be set for particles starting from early development. And I have just one last slide. If you've played Monopoly, you know this card called Chance, which says you can use this card to get out of jail free. Inherent particles, sometimes I hear people say, oh, I have inherent particles, so I'm okay with inherent particles in my product. Inherent particles is calling your particles inherent, and therefore saying we don't have to control them is not a strategy. Inherent particles is not a get out of jail free card. With that, I would like to thank you and give it back to Australia for. I would also like to acknowledge my colleagues at Lonza, Roman, Susanne, Athanas, and Hans Christian for the support of the work presented here. And I thank you on behalf of Lonza Drug Product Services.
Thank you so much, Dr. Singh. Now we will begin the question and answer segment of the webinar. Once again, I would like to remind the audience that they can still send their questions via the questions tab located at the right-hand side of the screen. The first question is, how can one choose between USB 788 and USB 787?
Okay. Well, part of it is obviously based upon the strategy. If you're going to multiple jurisdictions and you want to just stay with, not having to qualify and validate smaller volumes, then you can stay with 788. In case you have an extremely small volume product such as say an intravitreal injection, it can become, it can be a question of having to pool, say, hundreds of vials to get to the appropriate volume. and with that can become also difficult to, just the act of pooling may cause contamination and risk with the measurements, in which case it may be worth the effort to qualify a smaller volume method. So it depends a little bit upon your product strategy, your product characteristics, and the jurisdictions you want to work with.
Thank you. Are silicon oil particles acceptable, and are they counted in total counts?
So silicon oil particles are, from a safety perspective, are accepted. Obviously, they are applied to this, for example, to the syringes, and they are part of the product. But that doesn't mean you can slather on a huge amount of silicon oil and then just simply discount it. The product still has to be controlled. The siliconization process should be controlled so that you do not generate excessive particles from silicon oil. Again, the number of particles or the amount of particles you generate from a silicon oil should be understood and characterized, and then can be potentially discounted. But it does not, just because silicon oil is acceptable, does not mean that you have free-for-all in terms of not controlling silicon oil.
Thank you. What are the limits if using microflow imaging for QC testing? Are there any intentions to define such limits by the regulatory authorities?
There are no specific limits that will be identified for microflow imaging. There are no universal limits. I think these will have to be, if a sponsor intends to use flow imaging microscopy, they need to develop their own limits. And again, this goes back to what I was talking about, that having a proper particulate strategy so that you understand the number of particles you have in your product, what has been characterized in safety and efficacy studies, and based upon that, develop particulate limits, just like you would for any other characteristic, such as aggregation and so on. But there are no universal limits that can be identified for this, and I do not believe that that's the intention either.
Thank you.
And in most cases, you would want to be able to set your own limits and not be beholden to some universal limit.
Why is membrane microscopy not allowed for protein particles in 787?
So I shouldn't say it is not allowed, it's sort of discouraged. The Japanese Pharmacopoeia actually doesn't have it. So proteinaceous particles are harder to detect and analyze on a membrane. I realize that there are newer developments in this area that may improve the way this can be done, but in general, if you understand, even if you saw the pictures that I had for proteinaceous particles, these are gel-like amorphous structures, and filtering them, they can change their shape, their size on a membrane. It's just considered not as reliable as, say, light obscuration. So that was the reason for discouraging the use of membrane microscopy as the primary method in 787.
Thank you. How do you calculate particle density? Slide 42.
Well, I'm not an expert in this area, but refractive index matching is definitely one way. Sorry, you say this is particle density and not refractive index. Yeah, particle density. So we're talking about proteinaceous particles, I'm assuming here. Actually, there was some very nice work done by Hans Christian and that was published some time back, where they used actually resonant mass measurements, and just like you would do refractive index matching, they did density matching and looked at points where the proteinaceous particles sort of disappeared and became normally buoyant with the medium, that's a pretty nice way of calculating proteinaceous particle density, but I would say for the, to begin with, one can use values from the literature.
Thank you.
Unless you have some exceptional conditions under which you generate particles.
Is a harmonization between USB 789 and 787 planned? Currently, USB 789 currently only allows high volume light obscuration.
I don't know of any current plans for this right now. So let's say, well, if I was developing a strategy for an ophthalmic product, the limits would be 789, but you could do, just like as I mentioned, you could qualify a smaller volume method, and use a smaller volume method, but you would still have to apply the 789 limits to that. So I suppose in principle you don't really need harmonization. You're free to use small volume method based upon 787. Just apply the 789 limits, and of course you have to qualify the method.
You discussed MM for 787 testing. What are your thoughts about MM for 788 testing?
Membrane microscopy, I think there is no difference whether you're using the same concerns apply. Whether it's not 788 or 787 per se that's the issue. It is the type of product that you're measuring that is the question. If you're looking at biotherapeutics and you're working with proteins, it's the same concern around being able to truly quantify a proteinaceous particle applies, whether it's 787 or 788.
Thank you so much, Dr. Singh. Before I finish the webinar, I would like to ask our presenter, Dr. Singh, if you have any closing remarks.
I would like to thank the audience for listening, and my email is available, and I would welcome people to contact us at Lonza Drug Product Services if you have questions and other clarifications that you would require. This is an ongoing and evolving topic. As I mentioned before, a fair amount of skill and experience is required to work with these kind of methods and questions. So I would welcome people from the audience or others to contact us and if you have questions, then we'll request our services. Thank you.
Thank you. I would like to take this opportunity to thank again our presenter, for Dr. Satish Singh, Head of Drug Product Process Development in the Drug Product Services Group at Lonza Pharma and Biotech, for sharing his knowledge with us. I would like to remind our audience that they can view this webinar on demand by visiting biopharma-asia.com. Thank you so much, everyone. Have a great day.
Thank you.